السلام عليكم. روان السباعي. I did my PhD at UCL واليوم حأعطيكم محاضرة على the basics of neuroscience. Hopefully you guys تستفيدون. I'm really sorry if this is basic for some of you, but we want to build a strong foundation. I think these concepts are important. So we're going to start from the very basics and what is a neuron, how does it work, and then in the end we're just going to go through an example from a paper to kind of bring in all these concepts that I'm going to be talking about. Okay, so why do we study the brain? Some of you hope to understand or cure disease. But also the brain is pretty good at filtering and sorting information, and how it does that is a fascinating process. So whatever your reason is, I hope that you find this introductory computational neuroscience course useful. Okay, so let's start from the cortex, which is kind of split into these 4 different lobes, each of which is associated with a main overarching role. For example, the occipital lobe at the back here is used for visual processing, the cerebellum controls movement, etc. So this functional understanding that we have of the different brain areas and what they do comes from cases where damage occurs to these brain areas. The most famous case is that of Phineas Gage, who was a railroad foreman, and due to an accident, he had a metal rod go through his skull. Before the accident, it was said that Gage was a well-mannered, soft-spoken man, and he began to behave in an odd and inappropriate way after this accident. So such changes in personality would be consistent with kind of a loss of impulse control, which is known to be a frontal lobe function, and that's where the rod hit Phineas Gage. So although these cases kind of tell us a lot about how each brain region functions, how each brain area functions, more and more we kind of find that there is an overlap of function. So you can only guess why that happens. We don't really know. One thing, it might be like redundant information might be useful for processing and confirmation, or maybe even though we think the function is the same, there's actually a subtly different calculation that's going on that we haven't tested for, or it might be a bit of both, TBD. Okay, so the brain is an incredibly complex organ, and there's about 100 billion neurons. Each neuron receives about 10,000 connections, which, if you do the math, is a quadrillion synapses. So to put this number into perspective, there's 300 billion stars in this universe. So the brain is highly connected, there's a lot of talking going on, and these circuits give rise to complex behavior. Okay, so what is a neuron? A neuron is special because it's polarized, meaning that it has an electrical voltage across the cell membrane. We'll explain how that happens and what that means in a minute. Okay, so the neuron is composed of the soma with the nucleus where the genetic information is stored, same as any other cells. Dendrites at the edge here receive information and pass it down to the cell. The axon hillock is where the cell decides to send info or not, and it has the highest concentration of channels to help it do so. The axon, which you can think of as a wire, sends information on, and finally the terminal. So information generally flows from dendrites, cell body, to the axon, to the terminal. Okay, so let's go through the physiology of a neuron. The lipid bilayer arranges itself in a water environment where phospholipids face each other, and because of their hydrophobic ends. So this arrangement creates tight control of extracellular and intracellular ion concentrations on either side. In this way, it's said to be an insulator between two conductors. So at rest, there are more positive ions outside the cell than inside the cell, and this separation of charge across the membrane creates a potential difference between the exterior and interior of the cell, which creates an electrical gradient. So this is measured in volts. Because the cell can be charged in this way, it's called a capacitor. And how much charge a cell can hold is proportional to its size. Okay, so we talked about the diffusion of ions. So the diffusion of particles across the membrane releases energy. Ions generally move against with their chemical and electrical gradients across their concentration gradients. At rest, the membrane potential is closer to the equilibrium of potassium, and the concentration of potassium is maintained by this sodium-potassium pump, which exchanges three sodium ions for two potassium ions. And voltage-gated potassium channels move potassium out, whereas concentration is lower. This keeps the membrane at around -70 millivolts. So how easy is it for the ions to flow and diffuse across the membrane is called the membrane resistance. So most importantly, ions will continue to move according to these driving forces that I've briefly discussed, a mix of electrical and chemical gradients, until they reach equilibrium. So the chemical gradient is the difference in concentration across the membrane, and the electrical gradient is the difference of charge across the membrane. Okay, so I've explained three terms here: voltage, resistance, and capacitance. These three factors control ion movement or current movement across the cell. There is a relationship between all these terms that we have been using, and it can be described through Ohm's law, which is V equals IR. So V equals IR tells you that voltage is a direct product of currents times the resistance it has to flow through. So that in a cell with high input resistance, which is a small, i.e. like a small cell or a cell with fewer ion channels where it's not so easy for ions to move, there is a higher tension. So the current needs to fight to get in. And thus any current entering will create a bigger voltage step. Okay, so you can think of this like pushing water down an air hose. If the hose is small, you're going to need more pressure, and it's going to come out with higher pressure, which is the voltage. Whereas if the hose is a bit bigger, there's less resistance and it will flow more easily. So there won't be as much voltage. I hope that's clear enough. Okay, so we've talked about the movement of ions in and out of a cell membrane at rest. Where the cell is doing nothing, the membrane potential is more negative. So to initiate an action potential, we need to charge the membrane. So as the membrane gets more positive and depolarizes, certain types of ion channels will sense this change. These are called voltage-gated channels. As they open, they will let more positive ions inside the cell, mainly sodium. And the cell will fire an action potential. After that, potassium ions will move out of the cell to repolarize the membrane. So if you record from a cell, this is what that looks like. And each spike here is an action potential that a cell is firing. Okay, so let's move down to the terminal. Let's move down to after what happens after you initiate an action potential, what happens at the terminal. The terminal will activate voltage-gated calcium channels, and calcium will move in. And calcium is concentrated outside the cell and will move in across its concentration gradient into the cell. Calcium then activates vesicle transmission, and these vesicles are carrying neurotransmitters that will fuse with the presynaptic terminal membrane and release these transmitters into the synaptic cleft. So these transmitters will then diffuse across and activate receptors in the postsynaptic cell. So this is a chemical synapse. Generally, Dale's law states that each neuron releases one transmitter. However, we know that that's not entirely correct now. Okay, so now that you know how a neuron works, you can begin to understand how some of the tools we use in neuroscience work. For example, calcium imaging detection as a way to read neural activity. One technique I'm going to talk about is optogenetics, which work by manipulating ion concentration. So this was first discovered in algae, and channel rhodopsin permits non-specific cation influx into the cell and depolarizes the membrane. These channels are genetically expressed in neurons using AAV viruses, and they are expressed throughout the neuron cell, like the cell body. When shining light at the correct wavelength, these channels open and activate a neuron. So you shine a certain wavelength of light, the channel changes its the channel opens and allows certain cations in and activates cells. Okay, so one thing we should discuss is that neurons actually come in all shapes and sizes. And dendrites receive and integrate a wealth of information. In many cases, tens of thousands of synapses are formed on a dendritic tree of a single neuron. So generally, we call the ones that are further away from the cell body, we call the apical tuft, and the ones closer to the cell body, we call basal dendrites. These dendrites can come in all shapes and sizes as well. So activation of these synapses will result in a complex spatio-temporal signal integration involving current flow that ultimately converges at the axon, where decisions are made to either regarding action potential initiation. So what does this mean? Like when you're recording from a neuron, it basically means that if you record the voltage at a synapse, it will be much higher than the voltage at the soma. So the signal will attenuate as it gets to the soma. And this is called the rules that govern this is classical cable theory, which uses mathematical models to calculate the electrical current along passive neuroids. Okay, sorry. So basically, cable theory treats neurons as cylinders that are composed of segments and with capacitance and different resistance. So it doesn't take into account how many synapses it's receiving. It doesn't take into account how many ion channels it expresses. It only takes into account the geometry of the cell. Okay. So what does this mean in terms of circuit computation? Like, how does that translate into biology? Okay, so as an example, in CA1 hippocampal neurons, the input coming in from the entorhinal cortex, which targets cells which targets cells at okay, sorry about that. I need to get a glass of water. Okay, so let's get back to our example. So as an example, in CA1 hippocampal neurons, preferent path input, which originates from the entorhinal cortex, targets CA1 cells and relays sensory information about the environment. This input synapses at the distal dendrite and therefore are not propagated reliably. So you can see here, if you measure the voltage at the distal dendrite where this input where this input synapses, the voltage creates a pretty high change in voltage, but as you record closer to the soma, this input is not propagated. Okay. However, if Schaefer's collaterals, which carry contextual information that is important in memory formation, targets more proximal regions of the neuron and have a moderate effect at the soma. The interesting part is that when these two inputs are activated together, the dendritic spikes can propagate reliably, and you get a much more pronounced voltage change at the soma. This creates a kind of gating mechanism for synaptic inputs and integration. Okay, so let's talk about how neural circuits are connected and what you need to form a neural network. Okay, so let's build a circuit together. Cortical circuits are mainly composed of excitatory neurons, often with strong recurrent connections and fewer inhibitory neurons that curb local excitation. The core function of inhibition is to provide balance by dynamically suppressing excitation to enable rich and rapid dynamics. Okay, so inhibitory neurons constitutes a small but crucial neuronal class in the cortex. So while these cells kind of comprise around 10 to 20%, but they're incredibly diverse. The most common type are somatostatin-expressing interneurons and interneurons that stand for the calcium binding protein parvalbumin. Okay, these interneurons also target different regions of the cell. And inhibitory synapses can be categorized into axoaxonic, so axon to axon, axosomatic, and axodendritic, based on their location of their synapse in the target neuron. So generally, we have interneurons targeting the soma and axon regulate firing, whereas those targeting the distal dendrites have a more complex role in regulating and integrating input. Also, there are specialized interneurons that target other interneuron subtypes. These are called vasointestinal peptide expressing interneurons, and they're shown here in green. Okay, so there are different arrangements by which these excitatory and inhibitory interneurons can come together, and we're going to talk about a few here. So the first one is feed-forward excitation, which allows one neuron to relay information to its to its neighbor. Long chains of these can be used to propagate information throughout the nervous system. Next, you have feed-forward inhibition, in which an interneuron is between two excitation neurons. This is a way of shutting down or limiting excitation in a downstream neuron in a neural circuit. Okay. Next, you have convergence or divergence. One postsynaptic cell receives convergent input from a number of different presynaptic cells, and any individual neuron can make divergent connections to many different postsynaptic cells. Divergence allows for one neuron to communicate with many other neurons in a network, whereas convergence allows a neuron to receive input from many neurons in a network. Lateral inhibition. A presynaptic cell excites inhibitory interneurons, and they inhibit neighboring cells in the network. This type of circuit is important in sensory systems to provide edge enhancement. Okay, and then we have feedback and recurrent inhibition. So in panel E, a presynaptic cell connects to a postsynaptic cell, and the postsynaptic cell in turn connects to an interneuron, which then inhibits the presynaptic cell. This circuit limits excitation in a pathway. Some initial excitation would be shut off after the red interneuron becomes active. In panel E2, each neuron in the closed chain inhibits the neuron to which it's connected. This circuit would appear to do nothing, but they provide a negative feedback loop that is important in motor movement, for example. Okay, last one, feedback or recurrent excitation. A presynaptic neuron excites a postsynaptic neuron, which then excites the same postsynaptic neuron. This type of circuit can serve as a switch switch-like function, because once the presynaptic cell is activated, this activation could be perpetuated. Activation of the presynaptic cell could switch this network on, and it could stay on. This panel shows a variation of this more complicated feedback excitation network, in which a presynaptic neuron excites a postsynaptic neuron that can feed back to excite itself. Okay, so these are the many different configurations in which excitatory and interneurons can come together. I hope that was useful. If not, we can talk about it during the discussion. Okay, so let's put together our understanding of neurons and interneurons and all these different concepts that we just learned in an example. So let's use CA1 as an example, because I've already talked about the preferent path, and we're going to build a circuit and give an example of a resulting behavior. So we saw that the preferent path from the entorhinal cortex synapses at the distal dendrite. This input is important in providing sensory information regarding the environment. So what you saw, heard, tasted, right? With Schaefer's collaterals, which target the proximal portion of the neuron, brings in memory and contextual information. This is information about what happened, when did it happen, how did it happen. Okay, one problem in this anatomical overlap is that how do you separate the information about the environment from whatever happened in that environment? So this is important for learning. Basically, you need to be able to generalize an environment. Okay, so for example, during aversive learning, such as fear conditioning, entorhinal cortex input provides information regarding aversive stimuli, and Schaefer's collaterals provide information of the fear memory. In this example, which is from a paper by the Sonzi lab, they designed an experiment to tease apart this problem that I spoke about, how to separate the environment from whatever happens in the environment. So they designed an experiment, and they basically trained water-restricted mice to lick small water rewards while head fixed on a treadmill. They exposed them to multisensory context cues, such as olfactory cues, visual cues, and auditory cues. Then they exposed and monitored their rate of licking. On day 2, they paired an air puff, which is uncomfortable to mice, with one of the they paired the air puff with one of the contexts. This caused a decrease in the licking rate for the stimulus that was paired with the air puff. So you can see here in this graph at day 2, there's the mean licking rate drops down with the cue that was aversive versus the neutral cue. Okay. So interestingly, some interneurons, which, if you guys remember, target the distal dendrite, which overlaps with EC input, were activated by this stimulus. And you can see here, they were imaging some interneurons, and they found that they responded to the air puff, but after inhibiting them, they no longer respond to the air puff. Okay, so this suggests that some interneurons may be able to compartmentalize information. So if you activate these interneurons, they no longer respond to this. If you inactivate these interneurons, they no longer respond to the stimulus, and the population activity of pyramidal cells increases, meaning that these interneurons can actually control the activity of pyramidal cells. Most importantly, when you inhibit these interneurons, this also had an effect on the fear response of animals. So basically, we go from if we inactivate some interneurons, which target the distal dendrites, resulting in an increase in population activity and prevented learning. So basically, this means that some interneurons do inhibit the input coming in and allows for learning. Okay, so this is my final slide. I've walked you through the basics of how a neuron works, what makes it work, and how you can put that together from circuit to behavior. However, it's really not that straightforward. 33 to 60% of the cells in your brain are not neurons. They're astrocytes that provide nutrients and immune cells that support neural function. Another thing you can also maybe be amazed by is that there are new cell types that are being discovered. Lastly, interneurons can also have long-range projections, which also have a variety of functions that can target either other interneurons or specific cell types. I hope that was clear enough. I'm excited to hear your questions, or if you have anything you'd like to discuss further during the Q&A. And thank you for listening.